like to invite Rebekah Evans from Georgetown University Medical Center, who will give us a talk on a similar, but not exactly the same topic. Related was the word I was looking for.
All right. Hi, everyone. Thanks for inviting or accepting my application to speak here. I'm glad that Sarah and I were able to give these set of talks together. This is really fun. And as Rafi mentioned at the beginning, I'm no longer a postdoc, but this is all work that I completed when I was a postdoc at NINDS in the lab of Zaid Khalek. So I'm going to be talking today about two different populations of substantia nigra pars compacta dopamine neurons. As a postdoc, I was really interested in understanding how these SNc dopaminergic neurons work, mainly because they're involved in action sequence learning, the initiation of action sequences, and because of their degeneration in Parkinson's disease. As Sarah previously mentioned, when at the time of diagnosis, many of these neurons have already degenerated. And if you lesion these neurons in an animal, you can recapitulate some of the cardinal symptoms of Parkinson's disease, such as slowness of movement or difficulty initiating movement. But what's really interesting about these neurons is that they're not all the same. They don't all degenerate at the same rate. So here's a schematic of the substantia nigra pars compacta dopaminergic neurons in a healthy human brain. And the neurons here are divided up into two groups. There's a ventral tier, which is shown here in these filled circles, and that's really a stripe of dopaminergic neurons that sits right on top of the GABAergic nucleus called the SNR, substantia nigra pars reticulata. Then right above the ventral tier is another stripe of neurons that's considered the dorsal tier. You can divide the dorsal and ventral tier dopaminergic neurons by the presence or absence of this calcium binding protein called calbindin. So the open circles here express calbindin and the filled circles do not. When you look at a similar schematic in a Parkinsonian patient brain, you can see that the ventral tier calbindin negative cells are heavily degenerated, but the dorsal tier calbindin positive cells in particular are still relatively intact. So what I wanted to investigate was what are the differences between these two populations in the healthy brain before any Parkinsonian pathology takes hold. And the reason I wanted, well, there were two reasons I wanted to investigate this. One, if we can understand what's different before there's any pathology, we could get a clue as to what is causing this selective vulnerability of the ventral tier, and can even start to develop targeted treatments that would either selectively modulate the ventral or dorsal tier, depending on the stage of Parkinson's disease that someone's in. The second reason that I wanted to study these two populations was because I really wanted to know whether they were redundant populations of dopamine neurons. Do they do the same exact thing? Do they process information in the same way? Or are they embedded in different circuits? So to answer these questions, I used a calbindin-Cree mouse line, and I bred that mouse with a TD tomato mouse line. So all the calbindin positive neurons in this brain are red. Then I was able to patch onto either cells that were red, calbindin positive, or dopamine neurons that were unlabeled, calbindin negative, and measure the electrical activity. So I do that with whole cell electrophysiology. And what happens is you bring a pipette down to the cell and you patch onto the cell and blow a hole in it so you can basically be electrically continuous with the cell and measure its activity. I also filled the cell with a calcium-sensitive dye so I could measure the calcium signal in the dendrites at the same time that I was recording the electrical signal at the soma. This is what that looks like under the microscope. And when I patched onto these two neuron types, I found at first that they didn't seem very different. When they're spontaneously active, dopamine neurons fire without any excitatory input coming in. They just fire naturally. Their action potentials were pretty similar between the two populations. They both had the same width and height action potentials, for example. But when I injected hyperpolarizing current and then drove action potentials to fire from hyperpolarization, which kind of mimics the conditions of the cell being under inhibition, then I actually found striking differences. So when I do that, if I just put a little bit of hyperpolarization in to mimic weak inhibition, just enough to stop the spontaneous firing, both the calbindin positive and negative cells would fire action potentials when I drove them to fire and then come back down to baseline. And the calcium signal in the dendrite, shown down here, was really pretty small. But if I inject more hyperpolarization, and which mimics stronger inhibition, then the calbindin negative cells ended up showing this long-lasting after depolarization that basically is a subthreshold depolarization that far outlasts the actual timing of the current injections I was giving, these action potentials here. And the calcium signal in the dendrite was really quite large at the same time. If I inject even more hyperpolarization, mimicking even stronger inhibition, both that after depolarization and the calcium signal in the dendrite was even larger. But in the calbindin positive cells, the same experiment didn't evoke these ADPs, or after depolarizations. Both the action potentials just decayed back down to baseline, and the calcium signal in the in the dendrite, was really pretty weak and didn't change depending on the level of hyperpolarization. So we wanted to know what this was due to. So we ran many pharmacological experiments watching on different channel blockers. And what it turned out to be was the T-type calcium channel. So when we put on TTAP2, a blocker of the T-type calcium channel, both the after depolarization and the calcium signal in the dendrite was greatly reduced. Really, the calcium signal was pretty much completely eliminated. And just to be sure that it really was a difference in T-type calcium current density between the calbindin positive and negative neurons that was responsible for the calbindin negative neurons having an after depolarization and the calbindin positive neurons not having an after depolarization. I did some voltage clamp recordings where I carefully measured the amplitude of the T-type calcium current in both cell types, and then normalized that by the capacitance of the cell, which really gives you basically how much current per unit area, or the current density that occurs in each cell type. And as we would predict, the calbindin negative cells had a higher T-type calcium channel current density than the calbindin positive. So we can draw our first conclusion here. Calbindin negative cells express a stronger T-type calcium channel than calbindin positive cells. Now, another interesting difference actually showed up when we hyperpolarized these cells, and that was in what's called the hyperpolarization activated cation current, or IH. When you patch onto a dopamine neuron, you can get an idea of how strong the IH is by measuring how well the cell comes back to a depolarized potential when you hyperpolarize it. So here you can see this peak that kind of dips down, and then the cell comes back just without any change in the current injection I'm giving. It's just the IH channel opening up. And you'll notice that both the calbindin negative and positive cells do have this voltage sag, but the calbindin negative cells have a stronger and larger amplitude voltage sag than the calbindin positive cells. And this means that they have more IH current. This finding has actually been observed before from some data from Jochen Roper's lab, and we also confirm that the calbindin negative cells have more IH. So that's our second conclusion here. Now, what's interesting about this combination of channels being different between these two populations is that even though IH and T-type channels are totally different types of channel, they both do a similar thing for the cell. So they both require inhibition or hyperpolarization to be activated, and they both help the cell come back to firing after a period of inhibition. So you can actually see that pretty well in these traces. The Calb-neurons negative cell here comes back to firing really quickly and even is firing 2 action potentials right here, right after the hyperpolarization is released. But the Calb-neurons positive cell actually takes a long time to come back to firing. And when it does, it just fires back at its normal rate. So I did want to quantify this over all the Calb-neurons positive and negative cells that I patched. And so what I did was I just hyperpolarized the cell and released it and measured how long it took for the first action potential to occur. When I do that, I found that the Calb-neurons positive cells overall and very reliably had longer and longer rebound delays. That means it took longer and longer for that action potential to come back to firing, for the cell to come back to fire its first action potential after a period of hyperpolarization when the cell was very strongly hyperpolarized, or even when it was somewhat weakly hyperpolarized, as in this minus 65 amount right about here. But the Calb-neurons negative cells came back to firing quickly, meaning they had short rebound delays, even when recovering from very, very hyperpolarized potentials. When I measured the calcium during this same recovery period of the Calb-neurons positive and Calb-neurons negative cells, I found that the Calb-neurons negative cells had much larger calcium influx in the dendrites when they were recovering from hyperpolarization than they did at baseline, and then they did then compared to the Calb-neurons positive cells as well. So because we found this difference, we wanted to know if these intrinsic properties would affect the excitatory synaptic efficacy in these cells differently. And by excitatory synaptic efficacy, I mean, how well does excitatory synaptic input generate action potential output in these cells? So I did the same experiment, but instead of just letting the cell come back to firing on its own, I stimulated with a stimulation electrode at 100 Hertz to activate the excitatory synaptic inputs onto these cells. And I had GABAergic blockers in the back, so I was only activating the excitatory inputs. When I do this from spontaneous activity, where the cells are just spiking along as they want to, both the Calb-neurons positive and negative cells were able to fire really nice bursts of action potentials in response to this excitatory stimulation. But when the Calb-neurons positive cell was recovering from hyperpolarization, that same exact stimulation, the same strength, the same amplitude, the same frequency, the same location, wasn't able to evoke a burst anymore. And it was barely able to evoke a single action potential in this cell. The Calb-neurons negative cells, on the other hand, were able to fire really nice bursts of action potentials, even when they were recovering from very strong, very high levels of inhibition or hyperpolarization. And that was true over many cells. If we measure the number of spikes that occur in this burst, when the Calb-neurons negative cells recovering from hyperpolarization, sometimes the number of spikes was even increased relative to the stimulation's efficacy at a more depolarized potential. But in the Calb-neurons positive cells, the excitatory stimulation was really only able to activate, maybe 1 or 2 action potentials rather than a real burst. So this means that even if these two populations receive the exact same pattern of inhibitory and then excitatory input, they're not going to communicate the same action potential output. These two types of neuron fundamentally transmit and compute information differently. So the next and final question I want to address is, do these two populations actually receive different synaptic inputs or not? To investigate that, we injected chann into the striatum. The striatal neurons, which Sarah introduced so nicely, are inhibitory, and they're known to project to the dopaminergic neurons of the SNc. We used the p-dynorphin Cre line, which also allowed us to get a selective infection of the striasomes. In these mice, I cut slices and patched onto midbrain dopamine neurons so I could measure their response to optogenetic activation of these axons. Now, because I'm using the P-dyne-Cree line, I couldn't be also using the Calb-neurons-Cree line at the same time. So I don't know a priori whether I was patching a Calb-neurons positive or negative cell. But because I spent so much time figuring out that these two cell types had different electrophysiological characteristics, I can actually use the afterdepolarization as a kind of electrophysiological signature for the Calb-neurons negative cells. So I'm going to call them ADP cells instead of Calb-neurons negative because I'm not perfectly certain, with confirming with staining or genetics that these were Calb-neurons negative or positive cells. But I divided my cells into two groups, cells that had afterdepolarizations, and I'm only evoking it with one action potential in this case, and cells that didn't have afterdepolarizations. These cells should be like the Calb-neurons negative ventral tear cells, and these ones should be like the Calb-neurons positive dorsal tear cells. Then, if I measure the effect of activating the striatal axons, I can see how well they're going to inhibit each of these cell types. Now, if I activate, here's a 20 hertz train of blue light, and that's going to activate the striatal axons, having them release GABA and inhibit the dopamine neurons. You can see that that afterdepolarization cell was very strongly inhibited. The spikes were completely stopped, and the cell was greatly hyperpolarized. While this cell that did not have an ADP, it really didn't have strong inhibition from the striatum. This was true over my entire population of cells. There was a significant difference in how strongly the striatum or striosomes were able to inhibit the ADP cells compared to the non-ADP cells. So this really strongly suggests that the striatum selectively inhibits the ventral tier Calb-neurons negative cells. But I wanted to look at it one final way, and that was with calcium imaging. So here I'm using the DAT-Cree GCAMP6 mouse. So all the dopaminergic neurons have the calcium indicator GCAMP6 in them. And what you'll see when I play this video is that the dopamine neurons here are in green, and each of these are different somas. When this ring of light shows up, that's when I'm activating the striatal axons. And I'll play it again, but you can notice that some of the somas, the calcium signal dims, meaning they're inhibited by this striatal axons. And in some of the somas, there's not really much change in the calcium signal. We're going to look one more time. Particularly right here, there's some good cells that are heavily inhibited. And then when the ring of light goes away, the cells come back to their natural calcium levels. Now, I was able to draw regions of interest and measure the calcium signal individually for each of these somas and chart them by their location within the SNc. So here's the dividing line, this dotted line, between the SNR, which is the GABAergic nucleus that the dopamine neurons sit right on top of, and the SNc. And then I've divided the SNc into a more ventral and dorsal location relative to this dividing line. All these cells are color-coded by how strongly they were inhibited. The darker the color, the more strongly the calcium signal was reduced by striatal axon activation. And you can see that the cells that are more ventral were more strongly inhibited than the cells that were more dorsal. When I did this in over 400 cells, I saw a very clear trend when I measured the distance of each soma relative to this line, this dividing line between the SNc and the SNR. And some of the dopamine neurons you can see were right in the SNR, they were strongly inhibited. And then other than that, the more ventrally located the cell, the more strongly inhibited it was by the striatal axon activation. So again, this confirms that the striatum really selectively inhibits this ventral tier of SNc dopamine neurons. All right, to conclude, we have two types of dopamine neuron, the ventral tier, which are Calb-neurons negative. These are also the ALDH positive neurons that Sarah mentioned. They're really pretty much overlapping the Calb-neurons negative and ALDH1A1 positive. And these are the cells that are really the most vulnerable in Parkinson's disease and degenerate first. Then we have the dorsal tier calbindin positive cells that are more resilient. I found that the calbindin negative cells have a stronger IH and stronger T-type calcium channel than the calbindin positive cells. Sorry, the calbindin negative cells have stronger T-type and IH than calbindin positive. And this finding is really interesting for two reasons. One, it could give us a hint as to why the ventral tier is selectively vulnerable. In particular, the T-type calcium channels letting in so much calcium into these dendrites could really contribute to the kind of overload of calcium that's thought to exacerbate neurodegeneration in these cells. That, coupled with the fact that these cells don't have calbindin, so they don't have as much calcium binding protein to take care of the calcium once it's in the dendrites, really could contribute to strain on the mitochondria due to high levels of calcium. There's some interesting papers that have come out since I published this work. One in particular shows that a Parkin mutant cells in culture upregulate their T-type calcium channels, and blocking those T-type calcium channels protects them from neurodegeneration. So of course, future work is needed to really determine whether the T-type channels play any role in the actual selective vulnerability of these neurons, but it's a promising direction to look, I think. Then the second reason this is interesting, and this goes along with the embedded, the circuitry that these two cells are embedded in, is that these aren't going to be redundant populations. The loss of those ventral tier neurons early in Parkinson's disease really is not going to be compensated for by the dorsal tier, because they're fundamentally transmitting information in different ways. There's not, they're not doing the exact same thing, and the calbindin negative neurons are not even embedded in the exact same circuitry as the calbindin positive. They receive this striatal input, and the calbindin positive cells really don't receive much striatal input. So this means that these are really distinct populations, likely in their function, and more work using genetic lines of mice investigating these populations in vivo would be really exciting. The final thing that's interesting is that the striatum projecting to these ventral tier dopamine neurons could be controlling its own level of dopamine, because those exact neurons go back to the striatum and particularly seem to fill in the striaisms. So there's some really interesting future directions that could be taken up here, investigating the feedback from the striatum to the dopamine neurons and vice versa, and how they can dynamically influence each other. And with that, I'd like to thank my lab. As I mentioned, I did this at NINDS in the lab of Zaid Kalik, and I'd like to thank him for being a fantastic mentor throughout this process. And Sherry Zhang, Emily Tweedell, Manwa Zhu, and Jefferson Asensio all helped with various aspects of this project and are authors on these papers. And I'd like to thank the Brain Initiative and NINDS for funding this project as a K99R00 award. And currently, I am running my own lab at beautiful Georgetown University. And so if you're interested in this kind of work, or you know someone who is, in particular, looking at Parkinson's disease at this cellular and circuitry level, that's what I'm planning to continue doing in my own lab. And please contact me if you are interested in collaborating or talking further about these topics. And I'd love to take questions. Thanks.
Thank you, Rebekah. First of all, that's a beautiful shot of the area of Georgetown. Again, there's the Q&A tab at the bottom of your screens if you'd like to send in questions for either Rebekah or Sarah. First, let me start off. So are there currently like T-type calcium channel blockers in clinical trials for humans? What's the status of therapeutics like for Parkinson's disease in your area?
So I don't think that there are any in clinical trials at the moment, but there are some that are being investigated preclinically. There's a drug called zonisamide that is known to block T-type channels. It seems to do other things as well, but that's being looked at in a lot of Parkinsonian models and seems to have some therapeutic effects. There's not, I don't know of any that are currently being tested in clinical trials, but some some T-type calcium channel blockers are being currently used in humans in epilepsy as therapies for epilepsy. So there might be some that are already kind of, tested for safety and available that may make good trials for Parkinson's disease.
Yeah, so like maybe perhaps like drug repurposing later.
Right.
Yeah. So when the ventral tier of the substantia nigra, like degenerates, then what about the dorsal? Is there like a kind of interaction in terms of compensatory mechanisms, or like what happens to the dorsal tier?
Yeah, that's a really cool question. And I mean, I don't really have an answer, but I think that is a really interesting future direction. In some experiments in the future, I think testing the characteristics of the dorsal tier after the ventral tier has been degenerated to see if there's some way that they can be maybe taking over the role of the ventral tier neurons, or taking on maybe some characteristics that are like the ventral tier neurons. There's some studies looking at the molecule ALDH1A1 and showing that it does actually have some changes in expression in Parkinson's models. So it's not like these cell types are always completely static. They can kind of change their characteristics depending on the situation they're in.
So follow up on that. That was actually my third question anyway, but like now that you've done a little segue into my third question, so if there's like a kind of expression pattern that can be interchanged, how likely is it that, for example, like if somebody gets diagnosed with early on Parkinson's and before their neurons degenerate, if somehow there is I have no idea how to exactly change the expression of neurons, but like to convert a vulnerable into a resilient neuron to prevent further degeneration?
Yeah, that would be amazing. I mean, I think there's so many directions that research needs to take for this. I mean, one is really identifying early onset Parkinson's disease. I mean, that's its own very difficult problem. But I think if that was possible, yeah, that might be when a T-type channel blocker could have the most good. I think by the time it's, at diagnosis, when the symptoms are already really showing, it might not be as effective because it's not going to have as many cells there to protect. But if it could be identified early, I think, that a T-type calcium channel blocker might be effective at slowing the neurodegenerative process.
And a question came in from Dr. Bilal Mohammed. Is there any correlation between alpha-synuclein accumulation and the calcium channel in the substantia nigra compacta?
I don't know. That's a good question. I'm not sure if alpha-synuclein has been tested for changing calcium channel expression, but I do think that the kind of detrimental effects of alpha-synuclein really are enhanced by a high load of calcium coming into the cell. It's kind of, they kind of work together. And so I don't think that the calcium coming into the cell is, the thing that's killing the cells in Parkinson's disease. It's really something that's just making whatever is killing the cells worse.
Gotcha. If that is the end of our question and answer period, if there are no further questions, there's a further question from Ali Salapur. Has anyone done single cell RNA-seq on Calb-neurons positive versus negative cells?
Yes. So there's a whole kind of data set that does have dopaminergic and midbrain neurons in it. I think it's on the DropViz website, and you can see it doesn't have all the cells in the whole brain, but it does have Calb-neurons positive and negative dopamine neurons, and you can look at them through ALDH1A1 or other markers. And it also has other cells around that area, so you kind of have to go through a couple layers to get to the dopamine neurons, but there is a lot of data about the different RNA expression profiles other than calbindin positive and negative and ALDH1A1 positive and negative in that data set.
Thank you very much. We have less than 1 minute to wrap up. So thank you very much for attending, and thank you very much for giving today's 3P seminars. I don't know when we're seeing each other again over 3P, so please stay tuned via following us on Twitter at 3P seminars. Thank you very much for today.